We are used to the idea that if we get a bacterial infection, we take antibiotics and it's gone. But now this is rapidly changing. Bacteria are getting resistant to all antibiotics that we have. I'm Juana Diez, professor at the University Pompeu Fabra, and I would like to explain to you this worrisome phenomenon. Why bacteria are getting resistant to all antibiotics, how and what can we do about it? Watch this.
Bacteria are getting resistant to all antibiotics that we are currently using, and this has led to a global health crisis that is challenging our ability to treat common infectious diseases. Plus, interventions such as organ transplantation, chemotherapy, or surgery will not be possible anymore if there's not an effective antibiotics. Well, you are familiarized with humans. When humans have descendants, the progeny is born with a set of genes inherited from their mother and their father. But this is not true for bacteria. Bacteria divide by binary fission and thus make clones of themselves. So, if they do not have sex, that is, they do not recombine genetic material from two individuals to generate a new one, how do bacteria evolve? How can antibiotic resistance spread so fast in the bacteria population? Bacteria have developed the ability to acquire new genes from neighboring cells in their environment to generate genetic diversity. This is called horizontal gene transfer, in contrast to vertical transfer that goes from mother to offspring. If we think about it, this horizontal transfer is amazing. Imagine that you will like the nice voice of Adele. So she agrees and gives to you her black voice gene. And ta-da! Now you can sing wonderfully. So, although bacteria don't have conventional sex, they do have horizontal gene transfer mechanisms for generating genetic diversity. There are three ways that bacteria can carry out horizontal transfer: conjugation, transformation, and transduction. I know, you've already heard about it in previous courses. But here we will focus on the features related to the spread of antibiotic resistance. First, let's talk about conjugation with more detail. Transfer of plasmids via conjugation is the main driver for the spread of antibiotic resistance in the bacterial population. This is also the only mechanism of horizontal gene transfer that requires physical contact between two living cells and links their cytoplasms. This contact can happen in gram-negative bacteria through a pili, a hair-like structure that connects the cytoplasm of the donor and recipient cells, or in the gram-negative bacteria through an aggregation factor that gets both cells together. The DNA, plasmid, or a part of the chromosome is copied in the donor cell, and then the new copy is sent to the connected recipient cell. Due to their size, the transfer of DNA is most efficient for plasmids than for bacteria chromosome. Unfortunately for us, the genes that confer resistance to antibiotics are carried mainly on plasmids. Moreover, in contrast to the other horizontal transfer mechanisms, conjugation can also transfer DNA between different species. Again, unfortunately for us, many genes conferring antibiotic resistance are located in promiscuous plasmids, which code transfer systems that allow the transferring of DNA to unrelated species. Consequently, the plasmids carrying antibiotic-resistant genes can spread rapidly through a bacterial population. But there exist other ways that bacteria use to share genes. Imagine that your neighbor is dead, and you go to the funeral to give condolence to the family. You remember your neighbor had blue eyes, and you always wanted blue eyes. Easy, you take his genes and now you have blue eyes. This is what bacteria do, and it's called transformation. Yes, it sounds pretty macabre, but anyway, transformation is the ability of some cells to take up freely floating DNA from dead bacteria found in the environment. Then, the DNA is degraded or incorporated by recombination into the genome, generating genetic diversity. However, it is important to note that only around 40 bacterial species have been described to carry out transformation, that they are not always competent to take the DNA, and that within populations of these competent cells, only about 1% actually carry out the full process. Moreover, They're only able to take up DNA that is very similar to their own, not new genes. Consequently, in comparison to conjugation, the contribution of transformation to antibiotic resistance is lower. Transformation is one of the key tools used in modern biotechnology and has allowed major genetic engineering advances. How is this possible if it's so inefficient? Let's take a look now at transduction. Can you imagine that we can use flu to exchange genes? Yes, bacteria are infected by viruses too. They are called bacteriophage. They use these viruses to transfer bacterial DNA between cells. You will hear more about the phages in the practical work. There are two types of transduction. The first is generalized transduction, where the virus randomly chops up the host chromosome and accidentally packets chunks of the genome, transferring them to the new host cells where it might be integrated by recombination. The second mechanism is specialized transduction and results from the virus going dormant by inserting itself in the host chromosome. At some point in the future, it reactivates, cutting itself out of the host DNA, and sometimes accidentally taking some host genes with it. As antibiotic-resistant genes tend to be coded in plasmids, this kind of horizontal transfer is not as important as conjugation in the spread of antibiotic resistance. So, up to here, we saw how bacteria manage to pass the genes conferring resistance to antibiotics. But why this resistance spreads so fast? In the absence of antibiotic, the resistant plasmids do not give a selective advantage and they are fastly lost. Can you believe it? Why is this not happening? The appearance of antibiotic resistance happens naturally in nature. The key issue is that some human activities have accelerated its appearance and spread. The overuse of antibiotics in human health and in livestock farming, now forbidden in Europe but accepted in many other countries, have kept a constant selection pressure for resistant bacteria. Another important factor has been the inefficient measurements of prevention and controlling bacteria infection diseases.
So the two main danger actually is the use and overuse of antibiotics in food factory and in animals production, especially the fact that we use the antibiotics as a growth factors since ages in US and all over the world. It have been restricted in Europe since 2001. And the second one is the overuse in human health.
Now we are seeing infections with bacteria that are resistant to all the antibiotics that we have. So now we do need new antibiotics, and we need to evaluate these new antibiotics on their effectiveness and their safety. A key problem with the development, though, is the large investment needed to get new drugs to a patient.
Of course, there's a very important issue about the return on investment. So if pharmaceutical companies invest in oncology, in diabetes, even in HIV, these are more chronic infections, so they can get more return on investment. There's a larger market. So all these obstacles and hurdles have to be tackled so that we make sure that there is introduction of new antibiotics.
What can we do to fight antibiotic resistance? We are facing a complex problem that includes many interconnected factors. Interventions addressing only some of these factors will have very low impact. Therefore, if we want to reduce antibiotic resistance, there's an urgent need to coordinate a response to this problem from different angles. Research institutions and pharmaceutical companies will play a fundamental role. How can they do that? Well, by promoting research projects to discover new antibiotics and develop alternative therapies to fight a bacterial infection, such as treatment with plagues, probiotics, or immunomodulators to develop new vaccines.
Now, the issue is that in the last 30 years, investment in developing new antibiotics has really decreased significantly. And therefore, clinicians are very restrained at this point in terms of what antibiotics are effective at treating these types of infections. The link between biofilms and antibiotic-resistant bacteria is quite close in that biofilms, because of the presence of persister cells and being able to trade genes between each other, can often have an increased level of antibiotic resistance. And so I think moving forward, the type of technologies we need to develop and the type of research that we need to do needs to really focus on both of these problems together, biofilms as one distinct entity, but also as antibiotic resistance and how they interplay.
Bacteriophage preparations have been developed and used in several countries, such as France, Poland, and Georgia. However, the discovery of antibiotics has hindered the exposure of phage therapy to the rest of the world. Clinical use of bacteriophages in the West has been limited after discovery of antibiotics in early 40s and the later development of broad-spectrum antibiotics. However, as the world is dealing with increasing cases of multiple antibiotic-resistant infections and the emergence of superbugs, an alternative treatment for these problems is highly sought after. Phage therapy is one of the alternatives that may be an effective solution to this problem.
Another fundamental aspect to fight antibiotic resistance is to inform the population about the problems and actions that they can take. Here you have some examples. Take antibiotics only when the doctor prescribes them. Take only the pills prescribed. Do not give antibiotics to others. And don't take left pills from previous treatments. Wash your hands. Avoid direct contact with infected individuals. Keep an updated vaccination profile. On the other hand, physicians and pharmacists can take some actions too, like improve the preservation and control of infections in hospitals and clinics, prescribe and sell antibiotics only when they are really needed, prescribe the correct antibiotic.
After watching this video, you should be able to describe the horizontal gene transfer in bacteria, recognize the relationship between the gene transfer processes and the resistance to antibiotics, identify the causes of antibiotic resistance, analyze the different levels of action to take against antibiotic resistance. I hope you have enjoyed the lecture and now you are more aware of the antibiotic resistance crisis. Do you know what are the main current research strategies to fight against this health crisis?